So the background for this is that H2R is a gene-protein coupling receptor, and G-protein coupling receptors are said to be generally located in the plasma membrane. However, there have been many studies where the G-protein coupling receptors are in subcellular organelles, such as the endoplasmic reticulum, Golgi apparatus, and even the nucleus. To start off, the researchers wanted to show the unique nuclear localization pattern of H2R in human lymphatic endothelial cells. They also explored many other cell types, including human intrahepatic biliary epithelial cells, lymphatic fibroblasts, rat cholangiocytes, and rat mesenteric tissues. The biological question they asked was how does the binding of the histamine impact the localization of H2R? And also, how does the pharmaceutical blockading chemicals, how do they impact the H2R localization? And they also hypothesized that the binding of the histamine positively regulates the translocation of H2R to the nucleus. And the pharmaceutical blockade of H2R causes its translocation to be to a lesser degree.
Hi, my name is Brooklyn Barnett, and I will be presenting the methods and assays for my group. So, just for an overview, the experiment, they demonstrated the nuclear localization of H2R in lymphatic endothelial cells. They were they were also demonstrating the nuclear translocation kinetics of H2R in response to its ligand and the effect of this translocation on pharmacological blockades. So the reagents used in this experiment were histamine, dihydrochloride, H2R antagonist, cimetidine, triton X100, fluorescently tagged histamine, X-Flau, FL histamine, phosphate buffered saline. All of these methods were useful in collecting data because they were able to show the localization of H2R and the co-localization of H2R when partnered with G alpha S. It also showed the ligand-induced nuclear translocation as well as the effective H2R blockade on nuclear localization. Lastly, it showed the effect H2R being hindered by cimetidine treatment, which reduced nuclear translocation. So for the methods used for this experiment, they used cell cultures, IF staining, surgical isolation, which they euthanized male Sprague Dawley rats and excised their mesenteries to separate their mesenteric lymphatic vessels and their tissues, and they had to make sure they had to take precautionary measures to avoid fecal contamination. So they also use blast analysis, and they use this to compare amino acid sequences of H2R to nuclear localized GPCRs, which was previously defined in our introduction slide. Fluorescently labeled histamine binding assay, which LECs were grown and cultured between 80 and 90% with cimetidine, incubated, and tagged with the histamine solution. They used nuclear translocation kinetics of H2R in the presence of histamine. And lastly, they used statistical analysis. They used this to obtain a t-test of the variance between multiple comparisons in order to receive a p-value.
In Figure 1, we see the direct localization pattern of the nucleus. In 8.2, we see the localization of H2R in red, PROX-1 in green, and DAPI in blue. Figure B indicates the immunohistochemistry staining performed on rat paralymphatic tissue. The tissue was stained with an H2R antibody, and it shows its nuclear localization. And the close-up view simply shows the localization pattern. In Figure 2, we see the localization between H2R and its signaling partner, G-alpha S. In Figure 3, we see ligand-induced H2R nuclear translocation kinetics in LECs. The LECs, which are lymphatic epithelial cells, were affected by histamine at different times, and the line graphs are simply representative of the nuclear localization of H2R. Figure 4 represents the effect of H2R blockade on H2R nuclear translocation. In A, we see the bar graph representing the percentage quantification of histamine-induced cytoplasmic versus nuclear H2R intensity density between 0, 30, 60, and 90 degree groups. Figures B.1 and B.2 shows LECs treated with histamine and cimetidine. And for C, the bar graph represents the quantification of the treatment of 60 degree histamine and cimetidine histamine group. And in figure 5, we see the inhibition of H2R by cimetidine treatment attenuating H2R nuclear translocation. Figure A is the control group, and with 8.2 as its line scan intensity graph, and B.1, we see LECs treated with the fluorescently tagged histamine molecule. In C.1, we see LECs treated with cimetidine and then with fluorescently tagged histamine. And in D, we see the entire figure 5 data, including the control, the fluorescently tagged histamine, and both fluorescently tagged histamine and cimetidine groups quantified into a single bar graph.
Okay, this is the interpretation of the results. Okay, the staining of PROX1, which is a lymphatic specific marker, H2R, and DAPI, which is a nuclear marker, demonstrate a nuclear localization of H2R in human primary LECs. Based on this, researchers looked at other tissues. In this, they looked at rat perilymphatic tissue, and the positive staining of this shows the same localization pattern as in cultured primary human LECs. From this result, researchers wanted to test whether G alpha S also exhibited nuclear localization with H2R. This is because H2R as a GPCR interacts with G alpha S in downstream signaling cascade, Figure B shows that G alpha S is expressed with PROX1. Figure A demonstrates nuclear co-localization pattern of G alpha S and H2R. And then figure C shows that H2R, it demonstrates again the nuclear localization of H2R. Lamin A is a nuclear envelope marker. So, researchers then wanted to see whether H2R would behave like other nuclear localized GPCRs, so they tested its response to histamine treatment at 4 different time points. The decreasing cytoplasmic nuclear intensity density ratio indicates that histamine treatment in LECs affects H2R nuclear localization. Based on these results, researchers decided to investigate the effect of pharmacological blockade in H2R in H2R nuclear translocation at 60 minutes. They chose 60 minutes because it was the point with the highest nuclear translocation in the last experiment. So in figure B, we see H2R activation is mediated by histamines, and HDR activation mediated by histamines is responsible for inducing H2R translocation, and figure B2 indicates that cimetidine inhibits H2R nuclear translocation, overall suggesting that the activation of H2R by ligands plays a key role in nuclear translocation kinetics. Figure 5. Okay, so researchers finally wanted to verify whether the interaction between histamine and H2R is involved in nuclear localization. And the increasing fluorescent intensity from our control group to the cimetidine and histamine group to the histamine group indicates that binding of H2R and histamines plays a key role in nuclear translocation events. And do we agree with the interpretations of the results? Yes, it appears that with the staining of the nuclear markers and H2R that there is H2R nuclear localization, and also it appears that histamine does have an effect on the nuclear translocation of H2R from these results.
My name is Ian Riling, and I'm going to go over the conclusions for this paper. So going into this, the researchers wanted to explore the nuclear localization pattern of H2R and how to regulate this nuclear translocation. So they wanted to answer 2 main biological questions. First being, how does the binding of histamine impact the localization of H2R? And the second being, how does the introduction of pharmaceutical blockading chemicals impact this nuclear translocation? Through their research, they determined that the binding of histamine causes an increased nuclear localization of H2R, and that pharmaceutical blockading chemicals preventing the binding of histamine to H2R caused a decrease in the nuclear translocation of H2R. So due to these findings, the researchers determined that the H2R histamine binding is critical to the nuclear translocation of H2R.
So what's next? The researchers could investigate how the histamine chooses to bind to the H1R versus the H2R receptors. They could also explore the impacts of H2R nuclear localization on lymphatic physiology. They could also look into whether or not there were chaperones involved in the H2R nuclear localization process.